The next speaker is Dr. Leopold L. Ilag. He also flew all the way from Sweden to be with us today, despite his fear of coronavirus and everything. Although his mom said, "Don't worry, you'll be okay." He is a homegrown Los Baños guy and goes home to LB every year. So we always see him every year. He got his BS Biology degree from UPLB and his PhD from Karolinska Institute in Sweden. He's currently a professor at Stockholm University and a visiting professor at the University of Lublin, Poland. His research interests are on proteomics, membrane protein biochemistry, structural biology, and bioanalytical chemistry. His specialties find applications in research with interest in the development and application of new mass spectrometric methods to address problems in the life sciences. Has been involved in the characterization of macromolecular complexes such as RNA polymerase, degradosomes, and ribosomes, as well as membrane proteins. He's also a Balik scientist, and we also have collaborations with him. He has established a private company which will produce and market his products, and so his private company funded his travel all the way to the Philippines.
Okay, so good morning, and thank you to the organizers, especially Dr. Raimundo or academician Raimundo, for inviting me to give a talk. I feel so at home with this audience because there are so many familiar faces, and it's just a joy to be actually in this community. Also because I realize it's a kind of a joint event with the Microbial Society of the Philippines, and I recall that my mom was a founding member of that, so I'm happy to contribute to this activity. So, I am, as was told, coming from Stockholm. So this is the beautiful Stockholm, and Francis was there recently. I was happy to meet him in the summer. And it's famous, of course, for the Nobel Prize. But most recently, I guess Sweden is in the forefront because of our beautiful environment and the concern for the environment, as kind of highlighted by Greta Thunberg. And so I feel a bit proud in a way that I'm actually, since the end of last year, was with the Department of Environmental Science and Analytical Chemistry. But since this year, our unit has moved to the Department of Materials and Environmental Chemistry. And in this department, So in this department, I am actually heading the lab for the biophysical and bioanalytical mass spectrometry. For this talk, I was advised not to go into so much detail in terms of the technical aspects, because I've eventually become more a chemist probably, although I've always felt a little bit insincere in that because I've been trained initially as a biologist in UPLB. But in any case, so I tried to touch on more diverse subjects because of the eclectic subjects that I personally am involved with in research. But because it has to do with omics and microorganisms, I try to factor all these into this talk. But please excuse if things are a little bit too diverse. But my point simply is to be able to touch on the topic so that we could have more discussions later. So it has already been expounded by most of the previous speakers up to the genomics, transcriptomics, and now it's my turn to actually talk about proteomics. Now, I've always been associated with working with proteins, but my field in mass spectrometry is actually on what is called native mass spectrometry, which is more a field where we look at intact things, so we don't actually break things apart that much. However, the classic proteomics is actually defined as the kind of like the elucidation of the protein complement in the system. So basically, as complementary to the genomics, what we're actually identifying are the protein products in a system, but it's very different from the genome in the sense that it's more dynamic. So you have actually a lot of changes that happens in time, and as a function of just the location and a function of the environmental influence. Although of course there's a dynamic aspect to the genome, but in our case for the proteome, it's a little bit more subjective in a sense because, or it actually evolves more because the expression of the protein is not always there. Okay, so your genes are always the genes that you have, but the types of proteins, and in addition to that, the modifications of the proteins create such diversity that it can be a bit intractable. And in the spirit of trying to introduce other omics, so here's just the reasons why you will have to do omics. I want to introduce people to probably something less known, and this is because it goes beyond even what is metabolomics. And this is what is called adaptomics. And it is interesting because it's a way where we use proteins, or specific proteins in our system, that are reactive to, mostly reactive to electrophiles. And the electrophiles could be these things here. And therefore, what happens is that the protein becomes like a reporter for how much you are exposed to certain things. Okay, so that's quite interesting. And it usually is on a reactive cysteine, for example, in this serum albumin protein. That leads to the ability then to actually do omics on how much you're exposed, and this has led to the term exposomics. And as you can see, it involves a lot of stakeholders, and I thought that was an interesting aspect to bring up in this forum, because it highlights how, when I was doing my science studies, I felt like very happy to do just my thing, but now I get closer and closer to having to deal with a diverse set of people, because it's not, your science affects a lot of other sectors. And I'll get into that towards the end of my talk as well, as we touch on this aspects of green chemistry, which I'll talk about a little bit. Okay, so the only technical slide that I want to highlight in the talk is the fact that one of the central strategies would be what is mass spectrometry, for my case, in dealing with all the omics that I have talked about. And in principle, this is actually easy to summarize simply by being able to put in any kind of biomolecule, or any molecule for that matter, as much as you could fractionate or separate into different subfractions, so to say, just to simplify the complexity of the system, and process it into sizes enough for the mass spectrometer to analyze. In this case, if you have proteins, then you have to digest them with an enzyme after fractionation, say with a gel, or sometimes with size exclusion chromatography. If it were smaller these peptides, then if you start with them, you could separate them with chromatography and so forth. And then these mixtures that you generate should be introduced into the mass spec, and there is a system of further separation. In this case, it's also liquid chromatography. And then the analytes that you produce here are ionized. And this is the crucial aspect when it comes to mass spectrometry, because you can only work with charged analytes. But regardless of where it came from, a solid, liquid, or gas, it's possible to work with as long as it's charged, either negative or positive. Okay, and then here is the mass spectrometer, where upon ionization, which is the front end of the instrument, and so this spectrum that you is simply a representation of what analytes were generated from the fragments, and if it were a peptide, for example, each peak here is identifying a particular fragment, and if you piece together all these fragments, you will be able to read out the sequence. Okay, so if you were reading RNA, DNA sequences by using sequencers, then the mass spec is doing basically the same thing for peptides and proteins. And for small molecules, you can reconstruct structures just by knowing the fragments. Okay, so so this is the heart of the instrumentation, or the approach that I'm expert in. Okay, So the next omics that I wish to then introduce as a result of this is the environmental omics, and because it was so basically environmental proteomics is what I'm most interested in, but all these aspects of the previous omics that were touched on factors in this, and I like to highlight the application on ecological functions in this regard to talk about the, or just highlight one of the things I've been involved with, and Andrew here, Andy Montesilia, and which is a follow-up, actually, a follow-up study to what Francis, I believe, with Dr. Lantican and Andy as well have worked on, which was more a genomics approach, but then we actually moved into doing the proteomics, because again, the idea is that if you do proteomics, you see what are really expressed there, not simply what is up or down regulated from the gene level. Okay? And so what is interesting from this work is because but Andy mainly, of course, worked with cellulose-enriched environments for the isolation of the, for the analysis of the metaproteome of the mud spring environment, okay? But enriching it so that you would get more what the microbial community are that would have cellulase activity. And this is very important that I'll touch on again later because of the importance of cellulose or cellulosic material when it comes again to green chemistry. So I brought up green chemistry again, which is going to be clearer later. Okay, and here is again just to show you the kind of workflow that he works with, or he worked with, or that we implemented, because this was actually a service that was done at the Karolinska Institute in Sweden. But what I would like to highlight here is the aspect that you use advanced instrumentation, but also the importance of doing computational analysis, as Francis also mentioned earlier. So these are the conclusions from his study, and what I would just like to highlight from this is that there is a limitation in the aspect of the sample preparation that has gone into the work, and this has kind of limited a little bit the extraction of what real results we could have ideally gotten. But that highlights the fact that the analytical process is quite important. Okay, so that just gives us that introduction. But now I shift gears to the work we have on toxins, and this is also something that gets elucidated further on with proteomics. But just to give you a quick overview in the few minutes I have. So this is an amino acid, a non-protein amino acid. That means it's not encoded by any, it's not incorporated into proteins through the normal gene. So you have, this is beta-methyl amino L-alanine. It's a neurotoxic protein amino acid, but it is actually something that was detected from the cases of ALS in the among the Chamorro people in the 1950s. And in the investigation of how it could have been caused, people have postulated it was because of the cycad seeds that were taken or eaten by these indigenous people from making flour from the cycad seeds. And this plant actually has symbiotic cyanobacteria in the roots. And these cyanobacteria were identified to actually have this toxic compound, but by analytics it was shown that the quantity that could be derived from here was not enough for the toxicity that was happening to the people. And therefore later they realized that because the flying foxes were part of the food chain, part of is a delicacy of the native people, there was a proven biomagnification, or maybe not proven, but it was postulated, a kind of like conjectured and a little bit supported, the idea that the fruit bat actually eating the seeds bioaccumulated the toxin, and it was this biomagnification that was responsible for the increased amount of the ingested toxin. Now, because I'm a bioanalytical chemist, what I was interested in was to actually investigate the accuracy of all these data that people were getting. And we were very much involved in these aspects of the analytical chain. And this aspect is mostly addressable by the proteomics aspect. And we were able, but these still require a little bit of, in the later part, the omics aspect still is ongoing for the determination of the mechanism of the disease. So basically what we have done is we've set up the methods that allow us to identify all the, this is the BMAA, and all the isomers, we were able to separate them and identify them and quantify them. So that was a big advance. So we were the first ones to give the accurate amounts of these compounds. However, the interesting thing here is the analysis of how it can be neurotoxic, and it is believed that it is actually misincorporating into protein. So what happens is, instead of, for example, here a serine tRNA, instead of being charged with serine, is actually charged with BMAA. So this is a way that the amino acid is misincorporated, and the misincorporation could lead to protein misfolding. And of course, protein misfolding is a well-known mechanism for neurodegenerative disorders, so that correlates well with the ALS. And in this case, we are actually interested in the analysis of this water flea, Daphnia, which is a sentinel organism, which is a model microorganism for ecotoxicogenomics. And so we study these animals for how the misincorporation could happen. And so, as I told you before, with this mass spec method, we would be able to analyze structures of proteins. And when we fragment or sequence them, then we will also know if there is the proper misincorporation. No one has shown the direct misincorporation. Okay. Yes, and so this was the work of my PhD student from some time ago, and it's been followed up by, so here's the isomer study, and then we found that the BMAA was not only found in the cyanobacteria, but also in diatoms. And then we have the also the studies in rats and how it could really be involved in as a environmental neurotoxin. And then by improving methods for the detection, by using permanent charges, we were able to also get the ability to actually detect BMAA where conventional methods would not have been able to. Okay, and then we have, so in summary, we have been able to use our bioanalytical approaches to identify all the producers and then the sources of these, where you could accumulate the produced BMAA and as it reaches us humans, the consumers. And yeah, so the idea is that we could do the basic science and trying to understand how it leads to neurodegeneration, but also it is important that our methods are in place for actual environmental monitoring, not only in the actual physical environment, but also in the, even in food, okay? So one thing that, as I wrap up, one thing that is sometimes missed when it comes to proteomics is that people think that once you've identified the proteins, then that's something that is the end in itself. But it's actually important to also see where they are in the organism, and this is where imaging comes in. And one of my former PhD students, who's now an established scientist in Uppsala University, has developed from our earlier work on permanent charges, has developed or has used an existing permanently charged reagent, a beryllium ion, which he could use to label some of these amine compounds. And for the neurotransmitters, for example, that he could really show that you could do imaging on the brain tissues. And this is very exciting because for the first time, this is using multi-imaging, for the first time it's possible to actually simultaneously measure all the neurotransmitters. And so he's started up a company himself based on this strategy. Okay, and in the last minute, I just want to highlight the future trajectory of this particular work. And we're trying to do the imaging on this Dacia model again. And there is a microbial, it's the microbiome, there's a suspicion that the microbiome of this organism allows it to tolerate toxins. Okay, so the investigation of this kind of ability of, or to identify what is in the microbiome, what is produced by the microbiome that allows it to survive. Exposure to toxic substances is one interesting aspect. And then we also work with further analysis of the food chain in the Baltic Sea. And most interesting for us now is actually to understand the aerosol transmission of BMAA. And this is just the instrumentation that we use. I won't go into the detail there, but this was started to be explored from the thesis of my former student. And these are two other former students who, just to show you that we have advanced into also looking at, for example, amino acids in the environment, in the atmosphere, and we actually look at the contributions of biomolecules in aerosol formation, and this leads to implications on climate change as well. And finally, I want to say that all of these kinds of advances require a feedback on the improvement of methods. So I hope you appreciate that it's one thing to have a goal, of course, to do something, but you have to know how to do it carefully, and sometimes that will require also the development of new methods. Okay, so with that, and I've run out of time, so I thank you, and I'm looking forward to your questions.